I would now like to introduce Stats National Biotech Reporter, Damien Gardet, who will be presenting today's webinar.
Thank you so much. Thanks, everybody, for joining us. As mentioned, we're going to look at some interesting ideas in the means by which we determine whether new drugs actually work better than old ones. So the agenda for today, briefly, we're going to go over what's so great about randomized controlled trials, the gold standard that has been upheld for so many years, and kind of delve into why they become so important, but also about some of the limitations tied to them in various approaches, and also why some new ideas have taken shape to find other, maybe better ways to determine whether drugs work. And then we're going to spend the majority of our time looking at a few case studies of interesting ideas that are outside that traditional paradigm of a randomized placebo-controlled trial. And then we'll save time for a Q&A for your questions as we go along. Okay, so what's so great about RCTs? We can abbreviate them. So probably a lot of you know all this already, and I don't want to bore you, so I won't belabor it, but I do think it's worth picking apart some of the attributes of RCTs, just because we're going to talk about some trials that deliberately modify them. So first of all, the phrase describes a study in which you recruit a bunch of people and then you randomly put them into one of a few groups. Let's say two groups for this example. Either they get some new medical treatment or they get placebo. So why is that important? First of all, the randomization reduces the possibility of bias. If every single person in the trial has an equal probability of getting the drug or getting placebo, then you can be fairly confident that your eventual result wasn't skewed by individual characteristics of individual patients. And then the controlled part is, perhaps the most obvious of the definition, and that's because the hypothesis you're testing in a drug trial is that getting your drug is better than not getting your drug. And the only way to be certain that you're really putting that hypothesis to the test is to make sure patients get placebo or whatever the standard of care is for a given disease. And then probably also important is the concept of blinding. And that means basically that no one gets to know which patients are on your drug and which are on placebo. And that also reduces bias from the people who are actually running your trial. The converse of a blinded trial is an open label trial in which everybody knows everything, and the results of those are often not taken as seriously or considered as rigorous. So here is an arguably convoluted graphic that maybe I should have chosen a better one, but I think it breaks down pretty simply what we're talking about here. You have at the top, it's basically a funnel. So anybody off the street who might fit into your trial gets assessed as to whether they're eligible according to the trial protocol, which is sort of the rules of engagement for it. Some people are excluded, and those who aren't excluded get randomized, which means they get one of however many interventions. And in this case, we're talking about drugs, and it's usually either drug or placebo. And the rest is pretty self-explanatory. In the end, you unblind the whole thing, and you figure out whether you've got a drug or whether you've done an expensive waste of time. But the RCT, despite being the gold standard, they're not always ideal. So a few examples of times where it may be impractical or even unethical to go through that gold standard route. In public health, let's say there's an outbreak of an infectious disease at a population level. How do you run a randomized controlled trial? Would it be fast enough to actually staunch the outbreak? And how do you stratify patients? Is it ethical to give someone a placebo vaccine, for example, knowing that you may have just doomed them to contract the Zika virus? Similarly, or I guess conversely, in rare diseases, let's say you've got a drug for a serious disease that affects about 1,000 people in the whole world. So testing it on all 1,000 of them is impossible, obviously, but testing it on 100 of them might be impossible, or at least vastly impractical. And then again, you have the issue of is placebo ethical? Let's say it's a fatal disease that can be diagnosed at birth. Can you give an infant a placebo that basically guarantees death for the sake of science? And then this is also conversely, there's the placebo effect. This goes the complete other direction. There's a clear and well-demonstrated placebo effect in lots of clinical trials, but it's particularly pernicious in drugs for the central nervous system. So if everyone agrees that it's there, including the FDA, and if standard clinical trials don't minimize that placebo effect, then there's a question, why are we relying on these standard clinical trials, randomized clinical trials, for new central nervous system drugs? And we'll get to more on that later. So another thing about, and this is a lot of numbers and I apologize, another thing about randomized clinical trials is they're really expensive and they take a long time. There's some numbers from Tufts that are sometimes disputed, but somewhere on the order of $2.6 billion in 15 years is what's required to get a drug from the laboratory to becoming an actual product. And the issue is, actual large confirmatory trials are getting more and more complicated, which of course makes them more expensive. These are data from the FDA in terms of the number of procedures that are required in one of those phase 3 trials, and then each amendment, as you learn and change the protocol, adds about $535,000 to the cost of the study. And then sort of on a more philosophical note with respect to the limitations of randomized controlled trials, here's some data. These are the results of a randomized controlled trial that was published in the BMJ last month. The question was, when you jump out of an airplane, does wearing a parachute have any effect on whether you survive? And so if you look at this data, and of course this is vetted and published in a reputable journal, I think it's resoundingly clear that there is no statistical difference in outcomes whatsoever. Parachute or not parachute, if this were a drug, I guess I would say, if parachutes were a drug, I think we could say definitively that they would not win FDA approval for use in jumping out of airplanes. But of course, the point of this study was not to inform skydivers, but rather to illustrate a really important fact about RCTs. In this study, the planes were not in the air, which you probably would have assumed based upon these results. And the results remind us that the design of a trial can dictate its results, which is to say, it would be impractical to run a randomized clinical trial of jumping out of flying airplanes with and without parachutes, because no patient would consent to being randomized into the control group, for example. And so I think the message that the authors of this did, beyond looking to amuse people, is to drive home that we should not simply accept something as true because there's a randomized clinical trial behind it. Not all randomized clinical trials are created equally. So with that in mind, we're going to get into some case studies of things that are not randomized clinical trials, or at least not the ones that you're used to. But before we do, we have a word from our sponsors.
Great, everyone. We have a poll question for you. How many of your company's clinical trial processes would you estimate are completed manually with paper? And please select the answer directly on your computer screen. The first answer is less than 15%, 25%, 50%, 75%, or 100%. And we'll leave this open for a few seconds to give everyone a chance to participate in the poll. Great, and we're going to go ahead and close the poll. And let's go ahead and share the results. So it looks like we have, and the big winner is less than 15%, with a close runner-up being 25%. Great. Thank you, everyone, for participating in the poll. DocuSign will present on the results of this at the end of the webinar. And handing it back over to Damien to continue the presentation.
Awesome, thank you. Let's see if I can get the poll results. There we go. Okay, so like I said, we're going to look at some case studies now. And the idea here, I think, is that we went through the means by which a randomized clinical trial is not ideal, practical, or ethical in some cases. So now we're going to look at some clinical trials that illustrate a time when there was a medical problem, a need out there, and an idea for a treatment that might serve it, but the people involved realized that the randomized clinical trials of yore might not be practical, ethical, or ideal. So, number one, a drug called larotrectinib that was developed by a company called Loxo Oncology. The problem that they were seeking to solve is basically boils down to this. Most cancer trials pick a very specific patient population. Third-line triple-negative breast cancer, for example. So it's genetically defined, the tumor is in one particular place in the body, and things can get even more precise from there. They're stratified by tumor type and line of therapy, first line, second line, et cetera. So there's been a genomic revolution in oncology, and we have drugs that treat ALK-mutated tumors, EGFR-mutated tumors. But even precision medicine has been tested and approved in specific tumor types, the lung, the pancreas, the skin, etc. But what do you do when your drug works on a super rare genetic mutation, one in which stratifying a trial for just patients with skin cancer is maybe impossible, and maybe it would only round up 15 people, and the resulting FDA approval maybe wouldn't even be worth your time. The answer that Loxo came up with is what's called a basket trial. So the particulars of the science here, Loxo's drug targets tumors with what are called TRK fusion mutations. And they're very rare. I mean, the numbers differ here, but it's generally less than 1% of all cancer types, all solid tumors, have these TRK fusion mutations. So that, as I mentioned before, would make it exceedingly difficult to enroll a trial on just patients with lung or just patients with skin cancer. So what Loxo did is employed this basket trial thing. We can get a little more into that. They got 55 patients, all with a TRK fusion tumor, but with one of 17 different tumor types. There was no placebo control, because again, if you can only find 55 patients with a TRK fusion protein, dividing it up for placebo would probably be difficult and make the results hard to interpret. And also there was no blinding to this study, obviously. It was open label. So this was their endeavor to answer this problem. And it's also important to note that they're not looking at overall survival, which is, as the randomized clinical trial is the gold standard for trial, overall survival is the gold standard for whether cancer drugs work. Rather, they were just looking at response rate, which is to say, did the tumor shrink for patients who got the drug? And I think an interesting background about Loxo's endeavor is a few years ago, when Loxo sort of told the world about this plan, there wasn't a lot of skepticism about the scientific basis of this. Rather, I think people were concerned that the FDA just wouldn't go for the idea of approving a drug based on this trial design that we've just described here. So did it work? Obviously, the most important question, talking about a clinical trial. And the answer, as this plot illustrates, is yes, very, very much so. So what you're looking at here is the maximum change in tumor size across these multiple tumor types for patients in this trial. And the results there are on the right of the slide. We don't need to dig into it, but with a response rate that high, and it's important to note that these are patients with advanced cancers who've already received whatever treatment medicine has to offer them, Loxo began to look very clever for their approach. The drug was demonstrated safe, the responses were durable, and I mean, this chart is exactly what people in oncology want to see when they're looking at a new intervention. So fast forwarding, larotrectinib won FDA approval in November. It's indicated for, as we mentioned, any cancer type that has this TRK fusion mutation. One thing that's happened since then is that Eli Lilly paid $8 billion to acquire Loxo Oncology. And anyway, the forecast peak sales for larotrectinib are around $700 million from the powers that be on Wall Street. So what do we learn from the Loxo example? I think, primarily is that the FDA is willing to be flexible. The dogma of a drug must treat a specific tumor type, it must be tested in a placebo-controlled trial, et cetera. I think the oncology division of the FDA has long been perceived as the most forward-thinking and flexible, and I think the Loxo approval definitely burnished that reputation. One thing to consider, however, is that TRK fusion is a special case, and not every biomarker works like that. The cancer targets we mentioned before from yesteryear, ALK, EGFR, et cetera, companies in the past have tried pan-tumor trials in those states, which is to say any tumor type that has, for example, an EGFR mutation, and there haven't been successes there. So the Loxo approval was a milestone, but I think one should be cautious about predicting future approvals like this, pan-tumor type approvals. And then the last thing, and this is one of many things we'll touch on today that could probably be a webinar unto itself, selling a drug that works for patients that account for less than 1% of patients with cancer will be difficult. The cost of the medicine has already been the subject of news, and you could argue, if you wanted, that Loxo's decision to sell itself to Eli Lilly was an acknowledgement that a big pharma company like Lilly is perhaps better suited to the blocking and tackling it will take to actually make this drug, despite its scientific merits, a commercial success. Okay, so now let's talk about depression. We're going to talk about Alkermes' drug ALKS 5461, which I would love if it had a nickname, but unfortunately that's what it's called. So, as I mentioned, there'll be a problem that each of these things seeks to address. This one's pretty simple. Depression drugs are very difficult to develop. There hasn't been a new drug approval in major depressive disorder since 2013, and that is not for lack of effort. Traditionally, FDA has required 3 confirmatory trials in depression, and it would prefer that they be positive in their outcome. And the issue, what has bedeviled programs for decades, is the placebo effect, which is particularly challenging in central nervous system disease, particularly in psychiatry, and then if you dig down even deeper, it is particularly challenging in depression. So the FDA looked at data from more than 20,000 patients across 85 clinical trials dating back 25 years and found that basically placebo is becoming a better drug every year. There's a statistically significant increase in the placebo benefit observed in these trials over time, which stacks the deck against anyone trying to develop a drug for depression because while you sleep, placebo gets better. So the problem that, in this case, Alkermes was seeking to address is there a way around that? Is there a way to mitigate the effect of placebo, but still rigorously test your new drug so that you can eventually claim, if it wins approval, that it works? And the answer is something called SPCD. So SPCD stands for Sequential Parallel Comparative Design, which is kind of a mouthful, but basically it's a means of running a clinical trial to answer the question we asked before. Mitigate the placebo effect, but also still know whether your drug works. It was developed in 2003, by some researchers at Massachusetts General Hospital, who wanted to do exactly that. And it breaks down into 2 stages. The first one is pretty recognizable, which is that all your patients are randomized to either get the drug or to get placebo. But then there's stage 2, and that's when there's an interim analysis looking just at the patients who got placebo. The ones who are responding, which is to say the ones who are experiencing the placebo effect, getting a benefit from nothing, they stay on placebo. But the ones who aren't responding, whose depression has either worsened or not statistically significantly improved, they get re-randomized to either get the drug or stay on placebo. So it's almost like a subclinical trial. And we can look at how Alkermes approached that visually. And yeah, I mean, this basically just illustrates what I just said. You can see that placebo group, which is the largest group, it's a 2:9 randomization ratio, not to get too into the weeds. And so the theory here, I think it kind of goes without saying, is that you can possibly mitigate the effect of placebo responders on the final results. And this is a quote from the paper that appeared in Nature publishing these results. And basically, it's to give you 2 possible conclusions. Either your drug doesn't work, or you've got such an insanely large placebo response that, you know, it couldn't be overcome. But basically, the idea is to unstack the deck, to address the issue that we discussed previously. So, most important question, as before, did it work? The answer is sort of. So, on the SPCD side, that approach was technically a success. The placebo response in stage 1 was about 27%. But in stage 2, after those non-responders got re-randomized, it was down to just about 11%. So that's a 40% reduction in the magnitude of placebo response. And that would suggest that SPCD did its job for Alkermes. But the actual efficacy results of 5461 were problematic. So in 2 studies, 5461 had a numerical benefit compared with placebo on scores of depression severity. But the difference between that wasn't statistically significant, meaning you can't rule out that it was just random chance. There's a 3rd study in which the drug didn't significantly improve those raw depression scores from baseline compared with placebo, but it did beat placebo on the measure of average change. So Alkermes called that a success and called the totality of the data a success, but the FDA drew other conclusions, and we'll talk about that a bit later. But first, let's take a look graphically at sort of SPCD in action. So in these charts, these are 3 different clinical trials we just discussed before that Alkermes ran on 5461. The drug is in green and placebo is in gray, as you might guess. So what you want to see is separation between those lines. That would signal that 5461 is actually doing something compared with placebo on these measures of depression severity. So, like I said, this sort of illustrates that SPCD might not be to blame for what happened to Alkermes because, as you can see in stage 1, the lines are pretty close together, which is bad. And in stage 2, which is post-re-randomization, they're further apart, and that's good. So as we discussed, the change is a good sign for SPCD, but then the relatively small magnitude of that change is what it ended up being a bad sign for 5461. So cutting to the future, this all culminated in a very painful day for Alkermes. The FDA convened a panel of independent advisors to go over these data and advise, as their title suggests, the FDA on what to do with it. And basically, those advisors disagreed with most of Alkermes' conclusions with respect to these trials. The company had argued that the totality of the data across all of these studies demonstrated, or suggested at least, that 5461 was better than placebo. The advisors, however, pointed out that there were a few super responders in the trial, and they skewed the average observed effect of the drug, and that if you remove those outliers, then the drug looks pretty unremarkable. So they voted against approving the drug on that day. And now the FDA has promised to make a decision, yes or no, on Alkermes' drug by January 31st, and it is, I think, fair to say, almost universally expected to reject 5461, despite the totality of the data. So what did we learn from all this? I think throughout the advisory committee meeting and throughout the FDA's internal review documents for 5461, it was clear, at least to me, that the agency is open to things like SPCD. The FDA has repeatedly acknowledged and published things on the difficulty of running depression trials, and it knows very well that there are a lot of products approved for depression today, but no one would argue that the disorder is adequately addressed in our society or in the world. So it seems like the FDA is broadly encouraging of creative approaches. And then at the same time, I would argue that SPCD came out well in this whole thing. It did its job of reducing placebo response from stage 1 to stage 2. 40%, I think a lot of companies running depression trials would see that 40% number, and assuming they don't take issue with the data, would say, like, well, that's desirable for working in central nervous system diseases. So arguably, the problem was with 5461, and you could argue that this method could still work with another molecule. That being said, last thing, I went on clinicaltrials.gov just to see how many people are using SPCD. I only found 2 studies actively recruiting, employing this model. There were 17 others that were either completed or terminated, and there were a lot on the terminated side. So just empirically looking at adoption, it's possible that SPCD is not quite caught on in the minds of people in this field, and I'm not sure that the Alkermes case study will encourage them. Okay, so let's move on to our last case study in the field of rare disease. It is a gene therapy called AVXS-101 that now belongs to the people at Novartis. So again, there's a problem that needs to be addressed. As we've already gone through, the gold standard for clinical trials is a large placebo-controlled study. Hopefully it follows patients for a long time. We can determine whether something is safe and efficacious, some intervention is safe and efficacious for a disease. But what if the disease in question affects just 1,400 people? And what if everyone who has it will die, almost by guarantee, within 2 years of being born? And furthermore, what if it can be diagnosed at birth genetically? We need to know we need to know if drugs are safe and effective before we let companies sell them. But in cases like this, the question becomes how little evidence is enough? How small can a pivotal trial be? So for AVXS-101, another drug I wish had a more digestible name, the answer ended up being 15 patients. So we should back up a bit. The disease in question here is type 1 spinal muscular atrophy, or SMA. So as we said before, it affects just over 1,000 infants, and virtually all of them will die within 2 years of their birth. It's a progressive neuromuscular disease that just gradually robs people of their basic functions as life goes on, and eventually the basic function of breathing is among those that goes, and that's the end. So in comes a company called Avexis. They have a gene therapy that works, I'm simplifying here, but basically by providing a corrective copy of the SMN gene, and that is the gene that is defective in babies with SMA, and thus sets them on the path to this neuromuscular destruction. So that gene therapy had shown remarkable results in animal studies, and the time had come to test it out in humans. So here is a little bit on the trial design. 15 infants between about a month and 8 months old. 3 of them got a low dose of the gene therapy, which was meant to sort of test tolerability, and 12 got what the company considered the therapeutic dose. And this is important. There is no placebo control for reasons of access, ethics, everything we went over before. The only means of determining whether this gene therapy has anything to offer is by comparing it against observational natural history data of infants with type 1 SMA from the past. So to give you an idea of how it worked, I guess it's a fairly simple study, but here's the design of it. You have those 2 cohorts. They were followed for 2 years, which led to a publication in the New England Journal of Medicine. And then some were followed, or they plan to follow them for 15 years. I believe the oldest patient that they've observed is over 4 now. And the fact that that patient is over 4 kind of spoils the next slide, which is the results of the trial. After 24 months, all of the patients were alive. That would compare to about 8% in natural history studies, which, as we said before, the disease is fatal virtually for everyone at age 2. So that's an immeasurable benefit. None of them needed ventilation, which is to say assistance breathing. And 11 were able to hold their heads erect for greater than 3 seconds. These are data that kind of feel a little bit in the weeds in this context, but I think the important thing is compared with the natural history of SMA, type 1 SMA, no one had ever seen results like this. And the most common side effect was elevated liver enzymes, which in a fatal disease, scientists concluded was pretty tolerable. So one more thing, this sort of speaks for itself. This is the data by patient. So these numbers are based on something called CHOP Intent, which is a test that is made by CHOP, the Children's Hospital of Philadelphia, as its name would suggest. And basically it evaluates motor skills. It's fairly complex, but the point is the higher your score on CHOP Intent, the better you are at basic motor functions like sitting up and turning your head and, of course, most importantly, breathing. So this slide kind of speaks for itself, and it's pretty dramatic. The dark blue line at the bottom is what we know about untreated type 1 SMA. So motor function degrades until around 24 months, which, as we mentioned, is when the disease is almost always fatal. The sort of rainbow of lines that you see above it are individual patients who got AVXS-101. So again, just sort of goes without saying how dramatic those results are. So now what? Catching up to the future. So Novartis acquired Avexis for about $8 billion, somewhere within the history that we just described. And so now they are pressing forward with AVXS-101. The FDA accepted their application for approval in December. It's expected, well, the government is shut down right now. That could change, but last anyone asked Novartis, they're expecting approval in May, and then followed by approvals in Japan and Europe in the near future. I think for what we learned about this, I don't want to open a whole can of worms here, but you might recall the story of Sarepta Therapeutics, which is a company that ran a tiny trial with no placebo control and some debatable efficacy, and then took those data to the FDA for approval. Sarepta eventually won that approval for a treatment to treat Duchenne muscular dystrophy, but not without a lot of controversy. Famously, a reviewer within the FDA described the treatment as a scientifically elegant placebo. There were patient advocates pressuring for approval. There were people warning that the FDA would be opening a Pandora's box of drugs with flimsy evidence getting their way to patients and companies profiteering. The reason I bring that up is I think the story of AVXS-101 provides a really interesting and very stark contrast to that. For one, on the regulatory side, Avexis was engaged with the FDA throughout the process. The Sarepta process was marked by a lot of confusion and debate over who was telling what to whom. But two, I think Sarepta's case relied heavily on biomarkers. Those are measurable goals that suggest a drug is working. They are surrogates for what you actually want the drug to do in an observable clinical sense. Avexis was looking at survival. In the same way that randomized clinical trials are the gold standard of studies, actual survival is the gold standard when it comes to outcomes on a fatal disease. And so I guess all of which is to say is that Avexis, like Sarepta, ran a very small study with no placebo control, but if and when AVXS-101 gets approved, and everybody expects it to get approved, there will almost certainly be no hand-wringing or FDA mutiny or any of the things that characterize the Sarepta process. I think that's interesting. The last thing that could be the topic of a webinar itself is the commercialization issue. How do you make money on a product that is dosed exactly once and can only treat 1,000 or so patients? Novartis very famously disclosed that it did some math and decided that this gene therapy would be cost-effective at between $4 million and $5 million a patient. They didn't say they were going to charge that much money, but I think it was perceived as a sort of trial balloon of how much is this really worth, and then, as I mentioned, who pays for it, how, et cetera. In fact, we may actually do a webinar on that in the future. But that is the next fascinating thing. And it's interesting, I think, also that Avexis, not unlike Loxo from earlier in this presentation, sold itself to a big pharma company before the questions of cost, access, reimbursement, et cetera, would have been asked of it. It's much more maybe tractable to just focus on the science and then leave that to your wealthy friends in Switzerland. So what's next along these lines? A few clinical trials and /readouts / regulatory things that I think are worth keeping an eye out for in this context. One is Global Blood Therapeutics, which is working on a drug for sickle cell disease. What's interesting about this is in clinical trials, that drug seems to safely increase hemoglobin, which is the oxygen-carrying molecule in red blood cells. And the company believes that increasing hemoglobin is a biomarker for success in sickle cell, which specifically they think that doing so will reduce the frequency of strokes or crises that afflict patients with sickle cell. But we don't know that that's true. And the FDA has agreed to consider approving the global blood drug based on the data they have already, the data on that hemoglobin production. But there's no guarantee that the FDA will agree with global blood. They could end up in a similar situation as Alkermes. I think the outcome of the FDA process for this drug will be interesting, and we'll have read-throughs for sort of the state of biomarkers and how we think about some of these serious diseases. Similarly, in hemophilia A, there's a company called BioMarin with a gene therapy, and not unlike the Avexis gene therapy, this one has a pretty discrete way of working. It works by boosting a protein that leads to blood clotting, and it happens to be the protein that's lacked by hemophiliacs with hemophilia A, which is why they suffer from bleeds in the first place. Their blood doesn't clot. The interesting thing about this trial is that BioMarin has been consistently releasing data from an open label study. And the gene therapy seems to be working, which is to say it's boosting levels of clotting protein. The interesting thing, though, is that some patients have had levels of that protein that are well above normal. And as I'm sure you know, blood clots are desirable for hemophiliacs, but they can be not desirable when they can lead to strokes, for example. So the question is, are these patients at risk of overcorrecting? Are they at risk of having a stroke? Nothing has happened yet, but I think it's going to be fascinating to watch how regulators deal with this. A theoretical risk has derailed some drug programs in the past, but then the seriousness of hemophilia A maybe will allow BioMarin to get a pass. And then finally, Sarepta, we already mentioned them before. Similar thing on the biomarker angle. They have a gene therapy for Duchenne muscular dystrophy. And the idea is that it will boost microdystrophin, a form of a protein that, again, in theory, is a biomarker for improving the symptoms of Duchenne muscular dystrophy. But again, we don't know that. And this is an earlier stage program, but I think watching how it plays out will be fascinating because we're sort of in uncharted territory with the FDA. Okay, if I can figure out this platform, we will get to some of your questions. Cool. Oh, here we go. So, gotcha. Okay. One question, this is from Diane Ushner. I think it relates to the Loxo question. How can you be sure that the change in tumor size is not due to the placebo effect? That's a good question. I mean, it's underlying every open label study, one without a placebo group, and especially one that isn't blinded, is that. How can you be certain that you're isolating the effect of the drug from the effect of the standard of care? I think in this case, I don't think anyone would say in oncology that a placebo is necessarily shrinking a tumor, but patients get standard of care in addition to the trial drug. And so a thing that is often worth keeping in mind when you see clinical trial results in cancer is we don't know what necessarily the standard of care was for these patients before they enrolled in the clinical trial. We know how many lines of therapy they failed, but this applies to central nervous system disorders too. The care you get in a trial is often much better than you get, for example, in a community health center. So it's not impossible that some of the results we saw from the Loxo trial are due to things other than larotrectinib itself. So here's the question. So concerning vitally important therapies, FDA has a history of being flexible with surrogate endpoints that dates back to the beginning of the AIDS era. Isn't this relevant to your rare disease example? Very much so. And this gets to something that has been fascinating to watch play out over the past. I mean, now it's decades, but I think it's accelerated in the recent past, which is the rise of patient advocates, patient groups, and the fact that the FDA has begun to more and more take seriously their wants, their needs, and their views on drug development, which the FDA had previously been siloed. That definitely has its roots in the AIDS era. There's loads of literature on that I would recommend people examine, but it also touches on the Sarepta example that we mentioned, where there was something approaching a scientific consensus that that drug was problematic and probably needed a new trial, but the pressure from patient advocates who had nothing else for their children with this devastating disease definitely played a role in the FDA's thinking. In fact, that's not conjecture. Janet Woodcock, who presides over drug approvals at the FDA, said as much. On the more proactive angle, though, rather than just individual decisions on a drug, the FDA has embarked over the past few years on what they call listening sessions or learning sessions with patient advocates. And they do this when there isn't necessarily a single drug up for approval, but rather they just want their future decision-making on drugs for a particular disease to be informed by what patients actually say. And one thing I've heard, somewhat anecdotally, is that FDA reviewers have been surprised and have modified their thinking after learning what patients actually want. The gold standard is often changing the course of a disease, but sometimes in certain diseases, there's a symptom like itchiness or sleeplessness or whatever. And when patients are asked, what do you want? They'll say that first. And I think there has been a coming together, or sort of meeting in the middle a little bit, of experts at the FDA and the gold standards that they are tasked with employing, and then people who actually suffer from or care for people who suffer from some of these rare diseases, of kind of understanding that what's desirable in terms of patient outcomes when you hear it from patients is often not what you assume if you're just a scientist working in White Oak, Maryland. Let's see. Here's a good one. What role do you see for external or synthetic control arm usage in clinical trials? That would compare the treatment arm against data previously collected in standard of care or placebo. This has felt like a hurry up and wait concept for a long time. The idea of like the in silico trial, the synthetic control arm, has been around for a long time. Very smart people have said we finally have the technology, the data, et cetera. I've seen people on the regulatory side say we're open to this if we can see frameworks that could advance, but I haven't seen, and maybe this is my own ignorance, but I haven't seen this sort of like killer app for this concept. And I know the people at Flatiron Health, which was a data company that recently got acquired by Roche, put a lot of work into this and have, I think, realistic approaches about how practical it might be. But theoretically, there should be a future in which we know enough about how a disease progresses and how certain interventions work that it would obviate having a control arm in clinical trials. I just think that until there's a really, really obvious use case and it succeeds in like arguably a randomized clinical trial of that form of randomizing clinical trials, I think it'll kind of remain a theoretical thing, an interesting concept moving forward. Let's see. There's a question about the Loxo trial. Did it increase survival, or did patients die at the same rate just with a smaller tumor? The question is, we don't know whether larotrectinib increases survival. It was approved based on response rate data. That could be problematic. It's not impossible that we will learn, and Loxo and now Eli Lilly are following the patients for that trial. It's not impossible that we will learn that the actual overall survival rate is not as impressive as that those initial numbers looked. This is sort of the bargain that FDA has made in fatal disease, specifically cancer, which is that for these patients who have failed every line of therapy and have this very rare mutation to their tumors, the agency has decided that that's enough evidence to win approval. It's not without controversy. I think patient groups have cheered that. I think Dr. Scott Gottlieb, the commissioner of the FDA, is proud of that record. But there are people at places like Public Citizen who worry that by approving drugs based on this evidence, none of this happens in a vacuum, and drugs aren't free. So the Loxo drug is approved. It costs, I don't have it in front of me, I think it's in excess of $300,000 a year as a list price. If we find out in the future that it is not as lustrous as we thought, the system paid that money. People paid that money, insurance companies, Medicare, whoever is on the hook for it. And so, that's the balance that FDA has to walk. And it's something that I think Gottlieb and others have addressed in the past, but there's definitely a conversation to be had about what should the rigor be for these therapies when we know, even though FDA doesn't control drug prices, we know that they're going to be expensive and someone's going to pay for them. Maybe let's do one more. There's a question about the Avexis gene therapy. Did they only measure survival? What about quality of life? The CHOP INTEND score on, obviously they measured survival. It's the easiest thing to assess, which is whether a patient is alive or dead. But the CHOP INTEND score that they used was a measure of motor function. I mean, quality of life is defined in many different ways. There's the ICER group example, which often tends to include the concept of disability. And so the CHOP INTEND scores, the motor function that these infants were able to preserve, I think is a fair surrogate for quality of life. It is difficult to get an infant to fill out a questionnaire, obviously, about how they're feeling. But when the contrast is a virtual 2-year death sentence, I think that regulators will probably look fondly upon the data that they did show with respect to quality of life. So that is a wrap. Thank you for all of your great questions. Thanks everyone for attending this presentation. If you have any other questions or comments, you can reach me on Twitter. I'm at Damien Garde. And if you don't already, please follow Stat at StatNews on Twitter. I will now hand it over to Siona from DocuSign, who will conclude today's webinar.
Thanks so much, Damien. Hi, everyone. To close out the webinar today, I just want to talk about kind of the systems that are supporting all of these awesome clinical trials across the scale here. I think it's very clear that overall the challenges of driving success in clinical trials is a constant uphill battle. And if we think back to the poll in the beginning of the webinar today, we saw that a good number of you are estimating that less than 15% of your clinical trial processes are completed manually, which is awesome. But we did also see that 30% of you are dealing with 25 to 50% of manual processes still in clinical trials. At DocuSign, we believe that industry leaders in life sciences are building a completely paperless, patient-centric system of agreement in order to drive patient engagement and participation and make clinical trials successful. So, you know, the industry's faced efficiency pressures for basically its entire existence when it comes to clinical trial success. It can really be a deal breaker, right? So considering the perception and participation of patients that companies really face today are harsh, with a third of patients saying that they would not advise their friends to join a clinical trial, and that 80% of clinical trials fail to meet enrollment timelines. Up to 20 of them never enrolling a single patient. Some of you may be surprised to learn that in life sciences, an estimated 40% of R&D costs are linked to paper-based processes. We've seen a strong correlation between the challenges of patient enrollment and paper-based processes, and know that the impact of embracing technology in clinical trials can be really impactful in driving that engagement. Now, if we think about the current state of systems at many companies, the odds are you're dealing with a mix of legacy systems. So these are likely a mix of manual and digital processes that are creating a disjointed patient experience and ultimately can hinder employee productivity. Across these 4 stages of completing agreements, preparing, signing, acting on, and managing, we find there are multiple inefficiencies when relying on legacy systems, partially or wholly. Across our life science customers, we found that those costs are quite significant, from wasting $77 per agreement to losing 21 days on average to complete 1 agreement. We also see on average that companies have 1 out of every 5 agreements completed with errors, which means more time going back and forth to correct those errors, or even worse, the document remains incorrect and could put the company at regulatory risk. Whether you're completing patient consent, investigator onboarding, or site initiation forms, there are agreements everywhere. And with a legacy system of agreement, that means there are probably needless cost delays and risks everywhere. If we look at patient recruitment specifically, again, the top challenge is attracting enough participants. The reality is a very small percentage of cancer patients enroll in clinical trials, and as a result, half of cancer trials fail due to under-enrollment. This was a challenge that one of our customers, a large not-for-profit hospital, faced. The customer is a large hospital in the Western US with over 2,000 physicians. And their legacy system of agreement was the bottleneck in patient enrollment. It involved a 13-page packet of legal waivers and forms delivered via the US Post Office, or sometimes worse, interested patients had to brave the hours of LA traffic to physically come into the hospital and complete the forms. This isn't just about numbers and under-enrollment. This is about the patients that need treatment. As you all know, every trial that doesn't happen means that patients desperate to try a new treatment cannot do so. And of course, that a life-saving cure for many more patients is that much further away. This customer works with DocuSign to make enrollment in their clinical trial a lot easier and faster. They first used DocuSign to fully digitize their cancer registry process. So interested patients got an instant response via email with a link that allowed enrollment from anywhere at any time on any device. They followed a guided process for where to sign and what to fill in, all adhering to regulatory and compliance guidelines like HIPAA and 21 CFR Part 11. This increased trial accruals by nearly 5 times, improving likelihood that trials would meet their enrollment targets and achieve a 75% reduction turnaround time for patient registry and consent. Adding additional studies to the registry and consent process, this customer realized additional cost savings up to 46% per completed document. Moreover, they were able to link registry patients to studies 27% more rapidly than, of course, with manual paper means. This was a game changer for the oncology department. The customer later went further by connecting DocuSign with its clinical research systems Encore, which eliminated that need to rekey data and helped achieve the $46 in cost savings per document. So what you see is a massive increase in the speed, effectiveness, and cost efficiency of a business process, where in this case, the business is saving lives. We see leading life science companies implementing a modern, purpose-built system of agreement and seeing these impactful results of doing so. The DocuSign system of agreement platform is really that technology foundation that can help automate processes across all departments, from patient registry to chain of custody and legal. Implementing electronic signatures is really just the start of streamlining agreements across patients and employees. There's definitely more you can do to improve how you prepare, act, sign, and manage agreements. In summary, a purpose-built system of agreement is what will help companies meet patient expectations and ultimately get products to market faster. Thank you again for joining today's webinar. I'll hand it back to Sam, our moderator, for closing remarks.
Great, and that's a wrap. Thanks to everyone for attending today's presentation.